should have said this. Casey Soska, who's the scientific director for Play, was supposed to give the morning talk, but Casey was unable to be here. So unfortunately, you get to hear me again. And I want to thank the organizers for scheduling this particular topic, which is a thrilling, right, engaging topic in the middle of the afternoon, right after you've had your coffee break. So, but with that said, I'm going to try very much to have this, even though there is some sort of important nitty-gritty, we'll be focusing largely on the data management things that we've learned within the Play project, and I'll tell you, you'll look at the gory details there. But we're going to try to leaven it a little bit with some humor, and even some singing and dancing if you're so inclined at the end. And I'm not exactly kidding. It's I mean, it's really up to you whether you participate or not. All right, so let's move forward. So I want to start with a cautionary tale, and that will be self-evident why it's a cautionary tale about data management. Then tell you a bit about the hyperactive curation process that we have implemented for the Play project, and some of the motivations for that and so forth. And then at the end, try to encapsulate what I think some lessons we've learned on Play and some lessons I've learned in other data management related projects that I have been engaged in the last period of time. So, but first with the cautionary tale, I just realized I need to do something here to do there. Oh.
Okay. Now we're okay? Sure. Oh, we weren't being recorded yet.
Oh, no, we're being recorded.
Oh, gosh. Will someone bleep that out of the...
This is all saved for eternity. Oh,
Okay.
now. Ah.
I think I'm going to mute again. How about that? And we'll just deal with the room audio for the how's that?
Yep.
I thought it was muted.
I can mute you.
Please mute me.
You're good.
Just shut me up. Okay. The audio is on.
On B-cell function. I think that I could use the data for my work on pancreatic cancer.
I am not an oncologist.
I know, but I think I could use the data for my work on pancreatic cancer. Do you have the data?
Everything you need to know is in the article.
No. What I need is the data. Will you share your data?
I am not sure that will be possible.
But your work is in PubMed Central and was funded by NIH.
That is true.
And it was published in Science, which requires that you share your data.
I did publish in Science.
Then I am requesting your data. Can I have a copy of your data?
I am not sure where my data is.
But surely you saved your data.
I did. I saved it on a USB drive.
Where is the USB drive?
It is in a box. It is in a box at home. I just moved.
But can I use your data?
There are many boxes. So many boxes. I forgot to label the boxes.
Hello again. Thank you for sending me a copy of your data on a USB drive. I received the envelope yesterday.
You are welcome, but I will need that back when you are finished. That is my only copy.
I did have a question.
What is your question? You might find the answer in my article.
No. I received the data, but when I opened it up, it was in hexadecimal.
Yes, that is right.
I cannot read hexadecimal.
You asked for my data and I gave it to you. I have done what you asked.
But is there a way to read the hexadecimal?
You will need the program that created the hexadecimal file.
Yes, I will. What is the name of the program?
Cytosynth.
I do not know this program.
It was a very good program. The company that made the program went bankrupt in 2007.
Do you have a copy of the program?
I do not use this program anymore because the company that made it went bankrupt. Maybe you can buy a copy on eBay.
I have good news.
You again.
I talked to my colleague. She knew a person with a copy of the software.
Then why do you need me? Everything you need to know about the data is in the article.
I opened the data and I could not understand it.
If you have the program, you will find it is clear.
Well, I noticed that you called your data fields SAM. Is that an abbreviation?
Yes, it is an abbreviation of my co-author's name. His name is Samuel Lee. We call him Sam.
I see. And what is the content of the field called SAM1?
Ah yes, SAM1 is the level of CXCR4 expression.
And what is the content of the field called SAM2?
That is logical if you think about it.
What is the content of the field called SAM2?
I don't remember.
What about SAM3? Is there a guide to the data anywhere?
Yes, of course. It is the article that is published in Science.
The article does not tell me what the field names mean. Is there any record of what these field names mean?
Yes. My co-author knows what the content of SAM2 is, and SAM3. And SAM4.
Can I talk to your co-author?
I'm not sure.
I would very much like to talk to your co-author.
Well, he was a graduate student. He went back to China 2 years ago.
Can I have his contact information?
He is in China. His name is Sam Lee.
I think I cannot use your data.
You could check the article to see if what you need is there.
Please stop talking now.
The short version of this. Now we're good. Okay.
No.
Wait. Yes. Okay, good.
No.
Oh, that's, hang on a second.
Video that's going to be created about this next.
Oh, we're recording. We're making it now.
Just see Candace.
I am muted. Why am I still hearing myself? Any ideas?
If you click on,
Oh, hang on. No, that didn't do it.
Oh, the audio thing in your top menu bar? The speaker? Nope, the very top. The Chrome menu bar. Like up next to the
Hang
clock.
on, it's-
Yeah.
Why am I not seeing? Because now I'm.
Oh, there you go. Yeah, right there.
That is. Oh.
Can we turn that off? Oh.
That's why I can't figure out how I'm hearing myself. That's something different. Let's try that. No. Yeah, it seems like it's a very strange.
Oh, there we go.
Oh.
Output, mute.
Oh, okay. No, I'm still.
Okay, hello? No.
Are we still here? Okay, that's, okay, so we have to, I don't really understand why that happened, other than we just changed. Okay, well, too many audio inputs, too many video inputs, and we don't have a someone who's on the audio and video board and on their own. So anyway, sorry about that. What I'm going to be talking about for the rest of the talk is about what we call hyperactive data curation. And this is a process of curating data, and we have an extensive discussion of this in the following paper in the journal eScience Librarianship. So what is hyperactive curation? It is the idea of embedding data curation, the organization, cleaning, quality assurance of data within the data collection workflow. So actually this idea in the information sciences of active curation is something that has been well known, but what we've instantiated in Play, and largely for some pragmatic reasons because of the scale, is to embed curation in the process of data collection as we go along. I think the bottom line that I'm going to suggest is first of all, there's no free lunch, right? There's no free lunch. There is work that must be done to organize data in a way that other people can use it. And to use another trite phrase, you can pay me now or you can pay me later. So you decide where that work needs to go. And with hyperactive curation, we're distributing it across the project. So the idea is when we get to the end, it is largely done. That's the goal. And it involves rigorous and thorough quality assurance, which is we think is critical during the data collection, and also with curating the data with a specific sharing target in mind. So we have some idea of where it's going and what it will look like. Although you will see that this is an iterative process. I believe that what we're engaged in is something very similar to what people in the software world, software engineering world, would call continuous integration, which is that we're continually iterating and integrating our data in such a way that there's always some functional product that can be used, even if it's not the final one. And so the data I showed you this morning was part of that continuous integration process. Those were the survey data as of last week when I ran the workflow. Okay, so what is, I will ask you this. I will show you my workflow, right? If I show you mine, will you show me yours? Workflows, okay? You have a workflow. You may not have diagrammed it. You may not have indicated all the different steps, but you have a workflow in your research. And there may be parts of it that you're not sure about yet, but you have a workflow. So one of the things that we did on Play was we documented our workflow. And this diagram documents it to a fairly extensive degree. So including how do we identify the data collection and coding or annotation sites, collecting it, how that process works. And it's a flow diagram with labeled who does what, where, when. Well, when isn't really on here, although orderly, right? So we're going to go through over the next couple of minutes some of the details about this process. But the idea is going from the who's part of the study, the collection of data, down to, yay, we've got a Play data volume that we share with the research community that everyone can use. That's the ultimate goal. All right, so let's focus on the collection steps. So the collection steps for us involved identifying collecting sites and coding sites. Part of that identification process was embedded in the recruiting of various researchers and labs. And I would say one of the things that I don't know that we made this explicit, although we did mention it in the grant proposal, but in terms of the identifying people to participate in the Play Launch Group, Kathy, Karen, and I very much wanted to engage early career researchers who were at R1 institutions and those that were primarily undergraduate teaching institutions, people who were geographically diverse in terms of their locations. So we attended to in the recruiting of participant researchers in play to some of the dimensions of diversity that are important for scholarship. But that was a conscious choice, right? So these were folks that were invited to participate based upon a number of criteria. So, and the coding sites as well, folks who had expertise in different kinds of domains and were willing to embark upon this large-scale effort. All right, so we have now about a dozen labs that are in the field. And some of those labs were in the field before the pandemic restrictions. Some of them have just come back online in the last number of months as some institutions have permitted in-person research to proceed. There are places that are not in the field, and one of them would be Penn State, because until very recently, in-person research where one of the parties was unmasked, which in this case would have been the parent, was not permitted. And there were times earlier on in the pandemic where Penn State would not allow us to talk about vaccination status of our research staff. And we decided not to pursue permission to conduct research under those conditions. We did not feel that was consistent with the research ethics we wanted to embody. And there were various institutions handled in-person research very differently throughout the pandemic. And so that was a huge issue. But in any event, labs that came online undergo an extensive data collection process because play involves sending the standard protocol, sending a single researcher out into a home with a video camera and some other equipment and collecting those data. The purpose is there to be as unobtrusive as possible in the home environment so that the researcher doesn't become the target of attention extensively. So there's extensive data collection training. Much of it is video-based. Much of that training is online. So if you want to see what the protocol's about, we have those links. Once research teams have conducted, once they're ready, they go out in the field, they do a pilot visit. That data comes back. It gets participants are recruited, the data are collected, the data are uploaded to Databrary, and our lab staff, our central staff, review them carefully for quality assurance. As we look at the videos, were the video camera angles sufficient for video coding of the various coding passes? Can we hear the audio? Did the survey questions get asked? There's a whole extensive range of quality assurance. If a particular data collection doesn't go well, we provide feedback and we iterate. A session that passes then goes into the queue for being coded. The coding data, so part of what we're doing with Play is each individual collection lab has their own page on Databricks. It's called a volume. In the new system, it'll be called a project, but it's a place for data that are being collected and are uploaded. Those volumes are private to that researcher and our research team. They will be shared more broadly at a later date, but for the time being, they are private to the researchers. From those data then, we draw those videos and other files and allocate them to a queue for the various coding passes. Transcription is the first one. And that transcription, so the transcription pass that the central staff here in green conduct at NYU. We have coders who are English speakers who provide an English transcript for English-speaking environments and a Spanish transcript for families where there are Spanish speakers. Child, mother, obviously the 12-month-olds aren't saying very much in either of those languages, but they are making utterances, and we code when they make utterances. All right, so those undergo their own QA procedure and then enter a queue for coding. For the initial data release, we are doing all of these coding passes ourselves at NYU for the first, so those include communication coding based upon the transcribed utterances, gesture coding of various forms. We can look at if you're curious about the specific coding manuals, what the definitions of the behaviors are, those are also on our website here. Nope, that's the new version. They're here. And we can look at them in detail after, if you're interested at some point. All right, locomotion coding, emotion coding, and object coding. These are, in our experience doing video coding, it's best to have your coders focus on a small set of decisions and add multiple passes. You can think of these as layers of information. They're vertical in the data view spreadsheet, but I like to think of them timed this way. And so they are layers of information that we might add to any temporally dense time series. And a vision that we have for the future of a developmental science would be, with resources like this, lots of people can be adding their own annotation layers. Some of them by human coders, some of them maybe machine assisted, et cetera, covering different domains, different aspects of the phenomenon, so that we can then synthesize, right? We can see the whole elephant, right? But from its constituent parts. Once these coding passes, so we have a coding pass, 25% of the hour-long session, so there'll be 15 minutes, is coded by a second coder for reliability. Reliability, then files that pass reliability then or go through another additional quality assurance evaluation, and then would be ready to be uploaded to a place for and merged for data release. Oh, thank you. Okay, I'm going to pause there in case there are questions about these pieces of the workflow. Anybody has questions? Okay. So the quality assurance pieces have two different parts. One is heavy, the other is light. The heavy QA we're doing earlier in the training process when we're bringing people up to speed with the protocol. The lighter one happens once the teams have had several successful sessions and we're just cross-checking that things are looking good. We use an R Markdown document to generate a semi-automated quality assurance report from DataQuery. The code for that is here. Those are not public. Here's an image to show you what they look like. So we do quality assurance checks on the spreadsheet data. That is the DataQuery interface for putting demographic data, for file name checks, and do we have the site ID? Is there a participant ID? Is the length okay? All of these kinds of checks that are automated. This is a virtue again of using of curating to a particular target, and especially a target that has an API you can interact with. And so we use this internally to manage the data collection across the 31 sites, but right now it's about a dozen that are active. On the survey side, we export the data and clean the survey using the Kobo Toolbox API. And then that gets downloaded in a form, and we use a series of enumerated R Markdown documents that would look like this. This is just a screenshot from our GitHub repo. So we do this first, we do this second, we do this third, et cetera. And that part of the reason for that is to make sure we don't have data loss. Part of it is also some of these are computationally somewhat longer and will get longer as, you know, right now we have about 60 sessions, but we're going to have 10 times that many soon. And there's not a great way to, other than downloading the big file, to go through that. And these are constantly being iterated. And you can see we do things like we save them as CSVs. MBCDI gets split off from the other survey data, mostly because there's some unique characteristics we want with that. And we want to work with Mike and others to make sure an easy import and eventually into WordBank, et cetera. There's a separate process of removing identifiers so we can be explicit about which identifiers we want to remove, et cetera. Again, all of this is shared now, so you can see what we're doing, et cetera. And then the final CSV is uploaded to Databrary, and we can reread it back. So right now, the Databrary, the open API doesn't allow easy uploading, so we have to do that manually. But in the future, we hope that will automate that step. Will be great. So this is a figure from the paper that summarizes all the different resources we use. So these are the pieces of equipment: the video camera, a decibel, a recording meter. This is essentially an audio recording, but of course ambient sound levels, Kobo Toolbox, Databrary, and DataView. DataView is the coding tool. We're using Box as the working storage. I alluded to some limitations that Databrary has as a working storage environment, and so we're using Box for the time being for that purpose. Some of the quality assurance procedures are Ruby scripts that are embedded into DataView, where you can look for spelling errors, you can look for other kinds of concerns. We're making extensive use of our Markdown documents, code written in RStudio. There's a Python component to this as well for the data management program. And some of these are manual, some of them are automated or semi-automated. And so everything from share, and all the documentation is shared online with the exception of the detailed heavy QA protocol. Some of those reviews we're trying to keep private, largely because we look at this as a learning exercise for all of us, and we're very sensitive to having researchers feel embarrassed about their initial efforts to conduct the research along this protocol. In fact, we're making it possible that when you code another video, you won't know what lab it's from, right? So as to reduce any possible inter-lab concerns that might arise. Assessing the data quality, sharing it, and then the annotation pieces. So things that are in process. Well, so we have the full protocol is on our website. You can go there here, and the full data collection protocol is here, how we do quality assurance, et cetera. That's already here. We're considering migrating that protocol to the book down environment, which is really nice for, here's just an illustration of that. I mean, it allows you to search for things like, you can look for the emotion codes throughout the site. Right, here's the transcription, communication, emotion. Anyway, it's a really useful tool, and that's something that we're looking to in the future. Also makes it easier to export a PDF or a Word document. Many people like to have physical copies of the manual in their lab, right, to go alongside the other resources, and to supplement the videos. Individual level files can be exported in the chat format, and so our intention is that all of the transcripts will ultimately be shared with TalkBank. We've been working with Brian McWhinney on that. And then, as I mentioned, any aggregated MBCDI data for incorporation into WordBank. And then a key component, so the video said, do you have a guide to the data? Data dictionary. That is a first piece, I think, going forward. One of the things that we can all aspire to is having a schema indicating what is the measure, what are the constructs, what are the different levels, how does it relate to other schema. That makes the data more openly searchable and integratable across sessions, and those are things we're still working on. So, some lessons we've learned. So Michelle's paper about toward cumulative science has come up. Michelle also said that psychologists tend to treat other people's theories like toothbrushes. No self-respecting individual wants to use anyone else's, and went on to suggest that the toothbrush culture undermines the building of a genuinely cumulative science, encouraging more parallel plane solo game playing rather than building upon each other's directly relevant best work. So I'm going to suggest to you that psychologists tend to treat other people's data management practices, tasks, sometimes displays like toothbrushes in the same sort of way. Not always, but more often than than a cheeky, open, and reproducible developmental science advocate might like, especially someone who's those of us who are interested in building a more cumulative science. And so one of the aims is to, and I think part of that is talking about data management like toothbrushes is a bit about, it's reminiscent of talking about someone's personal hygiene, right? Or your, and so I think there are understandable reasons why we're reluctant to talk about it, or, we don't talk about, no. And so I have a copy, we can link to the video, and if you feel a need to get up and we don't talk about, we can do that. So I do have it queued here, so we were playing it earlier to test this out. So plan your work and work your plan. You have to curate your data for yourself. You have to. So what's clear to you is should be clear to others. Certainly clear to others in your lab. And whether you actually want to share your data or not, think about internal reproducibility with your own lab group. Can you give your data and workflow to someone else? You get hit by a car, you're out for surgery, or worse, and the science can continue, and they get the same result. That's a minimum standard that every single person in this conference can aspire to. Okay? And curating with a specific target in mind helps us think about where we're going and provides constraints on what is otherwise a pathway with many possible forks into it. Automate as much as possible. Script, use APIs when you can, exploit the web. Consistency is not the hobgoblin of little minds. It is the key to successful automation. By that I mean consistency in terms of your schemata, in terms of how you label your data, in terms of file names, in terms of names of the projects. These are the nitty-gritty details that will make your project go more smoothly or make it go less smoothly. And so there's no way to avoid, there's no free lunch. These decisions have to be made. And so agree, if you're working with a coalition, to make them and then stick with them. We've also found that we have to retest our workflow at every step. So it means we're looking at the data continuously, and when things aren't working, we revise and iterate. And rather than assuming that we're going to be done, so that diagram underwent many revisions and may undergo a few more revisions or workflow. We would expect it to going forward. We hope not wholesale overhaul, but it's going to undergo revision. Another trite phrase, so this is the trite phrase hour, don't make the perfect the enemy of the good. There is good enough for your purposes. And yes, time is short, life is short. We want to get science out, good science out, and keep that in mind as well. It may be helpful to you in talking to colleagues and others to know that meaningful data management plans are increasingly required by funders, and for example, will be required by the National Institutes of Health in January of 2023. And initially it looked like these plans were going to be kind of data management plan light, but the more I'm hearing from program officers, that is not the intent. And there are lots of folks who are going to be caught with their data down when this comes around. And so that may be a way to help you get the attention of some colleagues who want to attend to these questions about data management. So you're going to have to say, what kind of data are you collecting? Where is it coming from? How are you storing it? And where are you sharing it? And if you're not sharing it, why? And well, we don't have permission is not going to be sufficient, because then the question is, well, why didn't you ask? What is special about these data that they couldn't be shared? Well, there's no home for them. Well, here, so, and that may be true. There are not all data can be shared ethically. That's absolutely right. but the data for which, you are going to have to make arguments about why you're special and something else is not. And so ultimately, these things we think help us make data a first-class product, which it really is, right? It's the basis of our inference. It's how we know, it's what makes science different from lots of other modes of knowing about the world. It's that it's based upon evidence and logic and procedures. So that's our goal. So let's share best practices. Let's talk about, let's solve as yet unsolved problems together, right? Let's commit to adopting best practices, and let's not reinvent this. Anybody want to see the dates? Okay.